here. Okay. Awesome. Thank you. I thought I had it saved and then it was Bios GP instead of Bios support. Okay. All right. Bye.
Oh, yeah.
Oh, so audio is coming. No.
Mute myself. Okay. And we will
Share the screen as this. Perfect. And that one I put control, I guess. And
now.
allow, there's an allow for
Yeah.
recording, I guess.
So if you wanted to move that into a screen for.
Yeah, I tried and it didn't, but.
There. Oh, okay, sorry. I'll just close
Yeah.
all these.
Can you try moving one slide ahead to make sure the screen share?
Oh, it's not.
I
Okay.
saw the screen share got paused.
Yeah.
Then you go
Click
back.
on it. Cool.
Yeah, okay.
All right. I believe we're all set up. We're muted and ready.
And
Okay.
this microphone works?
Yeah,
Yes.
that's for
Perfect. Okay, hello. Sorry for the delay here, technical problems. Can everyone hear me okay? Cool. Okay, thank you. Well, so thank you very much to Professor Tinye. I know that she's not here, but it was an honor to receive this invitation and visit all of you, know more about your department. And I'm hoping that one day we can have kind of some joint seminars among the two universities and even other departments. I think we are close enough to start that interaction. So I'm going to work for that. Okay, so today I'm going to be talking a lot about where my research stands and about two projects that I have been working on. They are not, one of them is almost finished. Both of them are almost finished, but still adding new things to those to let them grow. So the title of my talk, Outliers or Reformists, it's more about the robust estimators. So it was inspired by an invitation that I got to ICOR's presentation. It's the International Conference of Robust Statistics, where they said, oh, you work in genomics data analysis, you work on regularization, and you work on robustness. Can you give a talk about the 3 topics? And I'm like, I work on all of them, but I don't know if I can give a talk on the intersection of those. And that inspires me to think about what is the role that robust estimation has in genomics data analysis. And I noticed, and that would be part of the talk. So I don't want to give you the answer to that. I'm very tempted to continue, but that is one thing. And then after doing that, I'm going to tell you a bit more about robust statistics and my research. Before getting into that, I want to remember and give this talk in memory of my mentor and friend, Professor Ruben Samar, that passed away last year. And he was a great inspiration for me to work in robust statistics. All that I know, or almost all that I know about robust statistics, I really learned it from him. And he was always an inspiration of my work. So dedication of this talk to him. I hope that he's also proud about the talk and he will not be upset with that. Okay, so I already say I'm going to talk a little bit about outliers in the genomics literature, the state of the art of the genomics, of the proteomics, of the robust linear regression literature, my contributions to robust regression, and where does it stand in the world of genomics data analysis. So this is what I was telling you, like robustness, genomics, and there should be another one about regularized methodology, which in a sense lies in the genomics proteomics part. Does that intersection really exist? So inspired by this question, together with my student, he's a current master's students in the Department of Statistics, we designed a regular expression syntax rule to detect terms of interest. And I'm going to talk about what are those terms of interest in different articles to try to quantify that intersection. That intersection feels pretty empty to me, but is it really empty? Is it really sparse? What is, can we do a quantitation about that assumption? So we basically use the NC-BI API to extract articles, metadata, and content. And based on that, we tokenize sentences to extract ideas from the text. We define rules in terms of if we see this world, this sentence is categorized like this or like that. So we went through that exercise and we refined those rules many times, depending on what we noticed that we were extracting from the text. And based on that, we start classifying and quantifying the terms that we were looking at. And we could put some numbers into this. The search that we did was from articles that are available in PubMed. PubMed, I think you all know, it's a database that contains many journals from medicine, statistics, and biological sciences in general. We look at articles starting in 2000, and the reason for the 2000 is because around the 2000 was the first time that the word outlier appeared in an abstract of those that we looked at. The word outlier appeared before 2000, but with a very different meaning. So we decided to restrict our search to PubMed articles, 2000 and up. And we found that there are about 4 million articles that are related to genomics and proteomics in oncology. We also restricted to oncological research because otherwise it's kind of too huge and we kind of got a bit distracted of what we were thinking about. And then only about 4,000 of those contain the word outlier in the abstract. But then we thought, well, outlier can mean many different things. So is this an outlier in the sense that we think of an outlier? So we went a little bit deeper and tried to look at the full text. sorry, before getting there, so this is how the word outlier appear across the different years starting in 2000. So in 2000 was kind of the first instances that we noticed outliers, and you can see with the years it became being more popular. Of course, this growth means also the growth of papers in genomics and proteomics literature, right? Because I'm looking at the intersection of those. So it's not only that outliers became more popular, it's also that genomics and proteomics became more popular, right? So I have a little bit of the two things here showing here. And then among those 4,000 articles, about half of them we could access the full text of the article, because we noticed that if we're going to talk about a very specific, robust method, we are probably not going to say anything in the abstract. It's going to be something that we will find in the method section or in the explanation or in the results or even in the discussion, but not in the abstract. So we need more than the abstract. And for that, we needed to look at about 2,000 articles now. Because we don't only want to see the word outlier, we also want to see what is the role now of robust statistics, given that you notice you have an outlier somehow in your data. So when we looked into that, we basically made another list of special terms. So a term that is very common when you think about robust statistics is the median. And that it's even something that we teach to our students in high school. If you have outliers, don't take the mean, take the median, because it will be more robust to outliers. So we took terms like the median, Huber. People think like, oh, I did Huber, so that means that I know something about robust statistics. Okay, so the word Huber, Tukey, median absolute deviation, estimators. So now terms like estimators means that you are really into more the core of robust statistics, more technical terms. Mahalanobis distance, trim mean, M estimators, outlierness, least trim square estimators. So some of them are more involved with robust methodology and some of them are more commonly used. And of those, we noticed that median, it's really what people are using when they are acknowledging some outliers in the data. Huber, it's kind of the next one being popular. But then what we consider more core, robust methodology does not appear very often. So we can see 75 articles talking about estimators out of the 2,000s that we have seen, okay? I don't know, I haven't explained too much this diagram, but the double dot means that, sorry, I don't have a pointer, that this term, so median and estimators appear in 16 articles, for example. So it shows you combination of the terms, but you also can see the median, how many times it appears, like the marginal counts, okay? Good. So from this analysis, we realized that robust methods seems to be outlying in genomic studies, okay? Now, with kind of, yeah, I mean, yeah, anyway, you understood what I meant. But now, they are outlying, but can they reveal richer information, okay? Even if they are outlying. So why people use them, or why should we use them? Why should we consider them? So of course, I work on robust statistics. So the answer to that is, yeah, you should use it. Yes, especially if you have outliers in your data, or if you believe you have outliers in your data. So now the space of a genomic study can be somehow represented by this picture, where we know we are in a high dimensional space, depending if you work at the DNA level, or you work at genomics level or proteomics level. And there's a lot of interaction among the features that you will be working on. So it's a complex landscape that inspire the kind of work that I do. Many problems in high dimensional biology can be modeled with regression data. Of course, they can be modeled with other things as well, not just with, and even without models. So, but what I work on is in how to use linear regression models to try to understand the the response that I'm interested in. So I'm looking at that regression model where I am assuming that the covariates are standardized and my response is centered. I have P regression parameters here and a normal error that are independent and identically distributed. And at one point, I will add outliers to that, okay? But for now, let's think about that classical model. I'm in a setting where P, the number of covariates, can be, and it's usually much, much larger than the number of observation, plus not all of those covariates are equally relevant. So I might be interested only in a handful of those and not the thousands and thousands that I'm measuring. So it's a very sparse setting. And on top of that, many of these covariates can be related to each other. For example, the genes can share the same pathway, and that makes them come in groups. And so should I get all the group, or should I select only one of them in the group? So I'm in this setting. Okay, so there are two different kind of questions that I'm trying to answer. One is, what are the features that are important and that there might be important for a biologist to try to understand a certain disease or condition, and how can I predict the response? So there's a variable selection problem and there's a prediction problem. In addition to that, what we know is that classical estimators will break down if they are outliers in the data. So can I make these estimators robust to outliers in the data? And finally, all this will happen in a very complex high-dimensional setting, as I showed you. So I want to tell you a little bit. This will become very heavy, but just that you understand a little bit where my research stands about robust estimators in general. So as I was explaining before, people have been working in penalized models, penalization, regularization models, trying to model high-dimensional data, trying to do variable selection, perhaps, depending on the penalty that they're using. And other part of the research literature is focused on robust statistics, trying to make methods robust to outliers. And lately, in the 2000s, 2010s, there has been an intersection of those two, doing robustness and penalization. But I also want to call your attention into, we can work in two different ways. And these are the two different ways. When we think about the least square estimator, you can think on the optimization problem that you're trying to solve. You want to minimize the residual sum of squares, or you can think of the closed form of the least square estimator, okay? So what people have done in robustness, for example, was, well, instead of minimizing the sum of squares of the residuals, maybe I can minimize another function of the residuals, okay? Or even maybe I can think that that is the variance of the residuals, and that is it's a function of the sample variance of the residuals. But why using the sample variance of the residuals? I can use a robust variance of the residuals. So some people have suggested getting to a robust estimator by minimizing another function of the residuals. On this side, if I think of the closed form, I also have here this is a function again of the sample variance covariance matrix. Remember that these are standardized. So this is the sample variance covariance of the covariates, and this is the covariance of the covariates and the response. So instead of using the sample version, why don't we use an S-estimator, for example, like a robust version of those? So this, in this column, we have what we call the plugged-in estimators, because we just replace in the solution a non-robust estimator with a robust estimator, even if they don't come from an optimization solution. On this side, we are just optimizing a different loss function, okay? When it comes to penalization, you know what is happening. We are adding a penalty here in those, but people have also been saying, well, instead of here, I can use a penalized version or a sparse version of the variance covariance matrix. And in fact, I'm going to give names to these estimators next, but this method, it's called SCOUT, and it was proposed by Daniela Wittens in your department and Rob Tipsirani. And then what happened with the intersection of these two? Well, here it's kind of obvious, like change the sum of squares by the robust function and just add the penalty. So that is what has been done. So another loss with the penalty, what happened here, okay? So I've been working in this last row for some time, and today I'm going to present to you what is missing here. Yes. Here? Yeah, no, there's not a typo. Perhaps it's just my lack of no, I didn't explain it very well, but I will in a couple of slides. Theta here, it's not the covariance matrix, it's the precision matrix. So it can be shown that this can be written in terms of the precision matrix. So it will be the inverse of that. And it's not obvious. You have to use the inverse of block matrices. And after a couple of algebra, you will get to this formula. But I'm going to show you in another slide better. Yeah, thank you. Okay, now before giving names to that, and just to make the landscape even more complicated, I need to say that there are two types of outliers. Usually people may think, oh, outliers, okay? And when they think outliers, they are usually thinking of this outlier. You're an outlier student. It's an outlier house, or it's an outlier patient. It's just a case, an experimental unit that is outlying for some reason in all their features. But in high dimensional data, we started looking at, or we started finding things like this, where if you think here that the columns are genes, for example, you may have one outlying gene. Another patient may have another outlying gene. And this is what we basically see. But it's not like everything in that patient is outlying. It's just some features that are outlying. And then the usual way that people lead with outliers is like, oh, just delete that case. Like, just ignore that one. It's an outlying patient, just ignore it. But if you start thinking that way for cell-wise outliers, you end up with no data, because everything has or almost everything can have an outlier here or there. So this was proposed in 2009, and in fact, Ruben Samar, that I was talking about before, was the senior author in that paper, where they proposed this new paradigm called cell-wise paradigm. And then the robust literature was also kind of divided into two. Are you robust to case-wise or row-wise, or are you robust to cell-wise outliers? Because methods that are robust to one do not appear to be robust to the other one, okay? So unfortunately, I needed to complicate it because there this distinction. So going back to that, and I'm not going to talk about all these methods because otherwise I won't be able to talk about my research. These are the methods that has been proposed in the literature. So as expected, there's a lot here in penalization and there's a lot in robustness, right? Like there are years and years of research in these two areas. Now, this area has also been highly populated because this one is the row-wise paradigm, the paradigm that we are most used to, thinking, well, we may have 10% of the cases are outlying. We know how to defend to those, okay? So a lot has happened here. The ones in greens are the ones that my group has developed. PENSEP and SEM come from penalized elastic net S estimators and penalized elastic net M estimators. And this is a multimodal one that if I have time, I would like to present to you today. And again, there's nothing here. So I want to talk about what we can do in this case here. I will go one more and then I ask you if you have any question. In the case of cell-wise, as you can see, well, this is the same as before, right? This has nothing to do with robustness, but you can see that there are not so many things because this is newer now, okay? There hasn't been so much research on that. And same for the intersection. And again, the ones in green are the ones that my lab is working on, and this is the one that I will talk about. So before moving into getting more deep into these ones, do you have any question? That I know that we usually do questions at the end, but I presented so much that, no, are we good? Perfect. Okay. Okay, so this is what I was telling you before. It can be proved that these things that you have here are part of the variance-covariance matrices, while this one, it's an estimate of the precision matrix. So basically, the way to get this equality is that you have to write your variance-covariance matrix into the blocks, one block corresponding to the XX, XY, YX, and YY. And if you use algebra on how to get the inverse of that one, which will be the precision matrix, you can get also these blocks on the precision matrix and prove this equality. Okay, so you can even see that there's a minus here. So it's not that obvious. It looks like, oh, this is the inverse, but it's done in a different way with the precision matrix. Okay. If we go the obvious way, this will be the precision matrix mixed with the covariance matrix, while this is all based on precision matrix here, okay? So as I said, Daniela and Rob Tirani published this in 2009, and they proposed this new framework where they used regularized precision matrix estimators to try to solve that penalization problem using the more the solution of the least squares, not so much the changing the objective function. Although to get this precision matrix, they have to go through an optimization process, okay? So the method, it's called SCOUT, and it's defined in 4 steps. In the first step, you are trying to estimate that comes from this optimization process. So it's the argument. So this comes from a maximum likelihood estimator of the precision matrix. So if you forget about the penalty, what you have here is the maximum likelihood estimator of the precision matrix XX, assuming multinormal variables, right? And then you can add the penalty to that so that you induce some sparsity, okay? They use P1 as an index there to choose the penalty. When P1 equals 1, you have a graphical lasso problem. And then in the step 2, they are doing the same for the whole variance covariance matrix. So now you have the Y, the response as well there, but conditional on the results that they got for the block XX, okay? And the reason for that is that they don't want to make the selection of the X's to be affected by the response. So they first want to see which are the variables that are conditionally independent, and then to see what is the relation of those with the Y. So they have that second step that is conditional on the first one, and then after that, they put the estimator together, followed by some rescaling. So this is, in very short, what SCOUT is about, okay? So the problem that you may catch here is that this depends on S. S here is the sample variance covariance matrix. And I'm sorry, I'm using S in another place for robustness, but S here comes from sample. And so if these steps depends on the sample variance covariance matrix, this is not going to be robust to outliers, okay? So some people have suggested that instead of using those S, you can use a robust variance covariance matrix there, and it will make GLASU robust to outliers. And these are some authors that have done that. So we thought, well, maybe we can do that in those steps. It's a little bit tricky because we have that conditional on also. So it's not so easy to do the optimization, but we work on algorithms that will do it together with 2 master students. They are now This one is already working on a PhD on her PhD, and this one decided to go to medical school, but we are still collaborating on this paper. We are working on a robust version of Scout this way. And I'm kind of going to go very quickly through these because we don't have too much time, but there are many options for robust multivariate covariance matrices. The one that we use is this one called DDC. And again, unfortunately, I don't have a lot of time for that, but what DDC is trying to do is to predict which are the cells in that multivariate space that are going to be outlying. And once it detects them, it tries to estimate or predict what the value of that cell should be if it was a robust process. And for that, it uses the data around the variable. Okay, so it's a long algorithm, it's a complicated algorithm, but it's based on detection and imputation. Okay, so we use that one, and in fact, it works also if you have missing data. So you can detect the missing data and impute it according to what should be the value of these if I trust the other variables around, okay? And it does it in a robust way. So we did that to get with Scout, and I'm going to present you here the results of this simulation study. We have more in the paper. The paper is still under preparation, but should be finished. And the dimensionality we are looking at here is 50 observations with 400 covariates, but not all those covariates are relevant here. Results are based on 150 iterations, and we have 3 blocks of 20 correlated variables, as this one is showing, and then the rest is all independent noise. Like none of these variables are related. It's just these ones that they come in block. Okay, so the true coefficients are 3 for the first 60 predictors and 0 otherwise. And they are related in blocks, as I said. And now comes the contamination. We have done both row-wise contamination and cell-wise contamination. I'm not going to explain exactly how we contaminated. If you have questions, I can go through that after. But we have looked at the root, mean square prediction error, and the F measure. And these ones are not robust because we are the data generator processes, so the test set does not contain outliers. So the idea is, can I build and can I train without being affected to outliers in order to predict a response that do not have outliers, okay? The F measure, it's the harmonic mean between sensitivity and specificity. And with that, I mean the variables that are truly related with the response, do I really select those? And the variables that I know that they are not related with the response, I have most of them are not related, do not select them, okay? So that is what measures the sensitivity and specificity. So the F measure should be closer to 1 to be good. The closer to 1, the better the performance. For the mean square prediction error, it's error. So the lower, the better, okay? The methods that we compare were different scouts. Remember that 0, for example, means no penalty in the first step that I show you. 1 means an L1 penalty in the second step. L1. So this is GLASU followed by LASU. This is more like a reach in terms of multivariate followed by LASU. This is a plain elastic net, a plain LASU. And then our robust counterparts with Pensée. Pensée is the robust counterpart of the elastic net, and sparse LTS as an alternative to LASU but robust. So these are the methods that we compared in the simulation. Okay, so in this graph, because I didn't explain how I contaminated the data, you have no idea what is K slope, but it comes from slope. So slope comes from slope. So it's we contaminated points in a direction. So case law tells you more about the direction in which we could put the outliers. So the bigger case law, the more damaging the outlier, okay? So I think for now, that's what you need to know. And in the y-axis, you have the rule mean square prediction error. And as expected, these are not robust methods. As we are more damaging with the outlier, the method starts to explode, okay? And the prediction error starts being very large. At the beginning, they are kind of okay because the outliers are kind of masking towards what is not an outlier, okay? And then when it comes to robust methods, you can see even the robust methods will not do very well at the beginning because they don't recognize the outliers. They are outliers there, but they still cannot act to be protected. So some robust methods will still do bad. But as the outliers become more obvious, the robust methods will perform better. This one here is one of the robust scout that is not performing very well throughout, but it's very consistent at least. So it's not affected, but it's not doing well, but it's not being affected by the outlier neither. Okay, so it's kind of being very consistent. We found overall that this scout 2-1 is in general doing very well throughout. Okay, now even if they seem to be slightly affected by that, even that one, when it comes to variable selection, we can see that the non-robust ones, the F measure will drop until everything is wrong. It doesn't select the variables that it should select, and it selects the variables that it should not select. They are completely broken. But when it comes to robust methods, you can see this one, the 1-1, that was not doing very well in terms of mean square prediction error, is doing very well in terms of variable selection. And the green one, that is the 2-1, is doing well in both selecting the features that should be selected and predicted according to what is expected, okay? Okay, when it comes to cell-wise contamination now, these estimators that you have at the very end are either not robust or they are robust to row-wise. So this is what I was telling you before, that row-wise robustness is not working well in this new cell-wise paradigm, okay? And the light ones here are the new robust ones that we design, and they are being robust to the cell-wise outliers. The classical ones are not doing well. And in terms of variable selection, these two are doing very well. This one is not doing that well. And these are the non-robust ones. And again, these are robust to case-wise, but they cannot deal with cell-wise contamination. So overall, this 1-1 or 2-1 seems to be doing very well. And I'm showing you only 2 scenarios, but we found the same in other scenarios as well. With that, I will pass to the last project. I think I have a couple of minutes, but is there any question about this project? I can also wait till the end for questions. Okay, so I'm really very excited about these projects. I want to show you a little bit, even if I don't go a lot in details. This is about robust ensembles. So ensembles now means that I'm not going to build only one model. I want to build many models, multi-models, and then I will put them together. But I want everything to be data-driven, okay? The variables that goes into each model and how I estimate the coefficients within each of those models. In particular, in this paper, we look at then for the prediction is the average of the prediction of all the models. You can use other measurements to integrate them, but we have used the average. So this is now our matrix of coefficients because we have multi-models, right? So in each column, we have a particular model. So if we think of a single model, as I was showing you before, it's only this column, only a single model, picovariates, okay? But I can also think of what is the coefficient of variable 1 across the G models. And maybe variable 1 is so powerful that appears in all the models, and I will have a value for that coefficient in all the models. Or maybe variable 1 will appear in only one model. So this will be a number where all these are going to be zeros. So how do I determine which are going to be zeros and which are not going to be zeros? Yes, Andrea. You're right. You're right. People understand different things by models. So let's say a regression function, a regression equation, a regression function. So I don't know if I have it, but I will show the objective function. But I think I put it here. Oops. Like many of these.
Changes it one model to the other. The outcome, the covariates are still the same, right?
Yes, the covariates are the same. These ones are the same. Yeah, but when you put them all together and when you allow to have more than one, maybe you prefer to say, let's estimate, let's do a very naive one. And let's say, what will be the prediction of Y if I only use the first covariate? And what is the prediction of Y if I only use the second covariate, right? Like you can do univariate analysis, and then you can say, okay, now I have all these predictions. What do I do with these? Well, just take the average of those, okay? So suppose, okay, let's go to a very simple case where we have 5 covariates, and the 5 of them are relevant. We know that they are relevant. People usually ask you, what should I do? Should I analyze them all together or should I analyze them one at a time, right? They do it with, for example, gender and yeah, let me show you the objective function. Yeah, let me show you the objective function, perhaps will answer to that. So this will be, I need to represent it in a way that I will allow to have the multi-model. So that's why they are in the matrix, okay? And I wanted to explain the notation first, okay? Let's see if it comes more clear later. By beta J, I mean coefficients of model J. So it will be the columns here, and beta G, sorry, and beta J dot will be the rows of these, okay? Now, the objective function that I'm trying to solve, and perhaps this answers more to your question, is this one. You see that the Y, it's always the same Y, okay? And the X is always the same X, but now I'm allowing this one to be a different kind, and then I have the double sums here, and I'm minimizing with respect to different things. And I have this subject to the different components here. Okay, so these first two controls the shrinkage that I will have within the models and the diversity that I will have between the models. And then this one here is going to say which are the observations that I will put in all the models. So the last one here is the one that will give me robustness, because I will not use all the observations. I will only use the observations that do not have a very influential residual, a very influential in the response to give me a large residual. So it's in the spirit of the least trim square estimator. Okay. I don't know if this became more clear, because yes, this one is the cardinality of IJ. Yeah. So I want to work with the IJ is the indices of the samples that goes into each of the J models. And this is kind of something nice about our estimator, that the samples that are used to estimate one model doesn't need to be the samples that are used to estimate another model, which is good because the outlier may not have, the outlier in the cell, well, this is a row case, but the outliers may not appear in the covariates that goes to one model, or the outliers will appear in the ones that go to another model. So by doing this in a data-driven way, the model can adjust better for this model, I prefer that this case is an outlier. For that model, this case may not be that outlying, depending on, this is a very high dimensional, so it's difficult to tell. So the model will take that into consideration. There are three other things that we need to tune the model on: the T, the U, and the H. Okay, think of a lasso. In the lasso, we have only one. Here we have three. So we are doing cross-validation to choose T, U, and V. And then, as I explained, we take the average of whatever is given in all those models to give a unique answer if needed to be a unique answer. Okay. Okay, I know it's kind of too much to get all of it from this slide. The paper, it's under revision. Hopefully it will get published and you can read all the details in the paper. But if you're very interested, let me know and I can give you a copy of the paper as well. There are some special cases here. If U equals G, this robust multimodal subset selection will be equivalent to the robust best subset selection, because we will have only one model instead of the multi-models. And then if T equals to P, this will become the least trim squares estimator. And if H equals N, then this is not robust anymore, and it's the best subset selection algorithm, okay? I think I said all that. Okay, so these are kind of the computational steps. I think it will be very difficult to grasp, but computationally, it's not an easy task because we have those three constraints that we have to work with. So we start by splitting the predictors. If, let's say, we need to have 10 groups, then we will, and this will be done through a grid, right? But if in the grid, we need to define what is the best solutions for, let's say, 3 models, well, the predictors are going to be split into 3 with these joint subsets. And then from there, we start optimizing and iterating until we converge. So we are using some subset block gradient descendant algorithm, and we prove the convergence properties of this algorithm. We use some local searches to optimize the way that we are searching for these solutions. And the algorithm has been written in C++. It's an R package already ready to use. And also in the GitHub of Anthony, you can find all the code if you want to look at it or if you want to replicate. It's memory efficient and it's been nicely designed. Okay, and so then finally I want to go back to a genomics application. This is not an application from my collaborators. It's a publicly used data set, sorry. It's been analyzed first by Schitz et al. And it looks at this protein, TRIM32, that has been correlated with this syndrome, Bardet-Biedl syndrome. And the idea in one of the papers is, can we predict the abundance of this protein with gene expression? And in fact, that is another part of my research that I've been working on, how much of protein expression can be predicted from gene alone. These 500 genes were chosen by the authors to be key genes in the problem that they were looking at. They have 120 samples. We have done 50 random splits where the training set is of size 50 and the test set is of size 70. And we contaminated 25% of the samples so that we kind of know what amount of contamination we are putting in each of these training sets. And then we repeat that, as I said, many times. And the results are going to compare some robust and some non-robust methods, some single methods, and some ensemble methods. In fact, in the paper, we added also random forest and a generalized linear model as well. So here we have elastic net, we have Pensée, we have Huber elastic net, sparse LTS. All these are single models. We have a robust best subset selection, that's a single model as well. And we have the robust multi-model subset selection. And as I said in the paper, we added other ensembles to compare the performance. The problem is that the other ensembles are not robust to outliers. So it's obvious that they are going to break down. And so we just put only one non-robust one. Okay, so these are the results. The results are relative to the one that is performing best. And we found that in all cases, the robust multimodal subset selection was being the best one. And the second best was the sparse LTS, which has, from the robustness point of view, is doing a very similar thing, is looking at only a subset of the cases, of the observations that are not affected by the outliers, but this is a single model and ours is a multi-model. And you can see by the standard deviation about the different splits that we have done, that is not always doing so well. There's a lot of variation, and a single model, even if robust, is not performing that well. And then same for other cases. And of course, the non-robust methods are not performing well when there are outliers in the data. Okay, so with that, I would like to conclude. In my research, I'm proposing various robust sparse regression estimators based on regularized multivariate methods or multi-models. We propose efficient algorithms, all of them written in R, and there are R packages that you can use for that. And all these packages are already published in CRAN. Robust Scout is robust to both row case and cell-wise outliers. The robust Scout show competitive performance compared with other existing methods. And I think I didn't put it, but the robust multi-model subset selection, as I presented it today, is robust to row-wise contamination. The cell-wise contamination, it's in production. And going back to this question, outliers or reformists, genomics and proteomics analysis data may contain many predictors. Only some of them needs to be selected. They are computationally very intensive. They are usually focused on classification, and the main objective is to predict a response. And robust estimators don't usually do very well in this task, or it's hard to do well in all these tasks. And I think these are some of the main reasons of why the robust methods continues to be outlying in that field. So I think that robust estimators needs to grow in this direction to have more presence in the field of genomics data analysis. Plus, we need to start including robust estimator in the standard curriculum if we want people to start using it, because otherwise they don't know about it. Even people in statistics may know about it, but when we do a collaboration with someone outside statistics, they don't know about it. The editors of the journal may not know enough about it. The reviewers may not know enough about it. So it becomes very difficult to cut that cycle. So the only way is to really add it into the curriculum, to start with education. I'm a big believer of education, obviously. That's why I work where I work. So with that, I would like to thank you all for your attention, for the invitation again. It's been a fantastic day, and I hope to see you soon in Vancouver. I want to thank all the other contributors of these projects. I would have not been able to do any of this alone. Anthony, Maggie, Charlotte, Javier, and all other members that they interacted very nicely, and they help each other to make all this happen. I was funded and I funded my students with NSERC grants and with CFI computational infrastructure. And if you want to learn more about me, this is my webpage, and I invite you to follow some of my work there. Thank you.
You know, and like, let's say you have a bus stop. How do you even analytical assessment, you know, of how well it works? Like, for example, you know, for nuclear, can you bound the prediction? Can you tell me how it compares to anything else? I mean, is this just, do I have to believe that this is just, you know, reasonable, that it works well in simulations, or is there any formalism that can?
I haven't done any of that, but I understand that. When people propose the methods, they basically show in simulation studies, in applications with control environments, but not so much to do a full analysis of the mean squared error. Most of the theory in robust statistics has been in the statistical properties in the sense of, do I have an unbiased, like, what is the consistency of the estimator? Do I have asymptotic normality of the estimators? In the ones that are coming now is, can I prove convergence? Will I end up where I want to end? But in terms of the prediction, what is the error? What can I expect for the error? Is it going to be completely unbounded, right, or not? The only thing that we usually do is to look at, which is not what you're asking, is to look at the breakdown point of the estimators, that it will look at a bound on the bias and know if it, but it does not what you're saying. It's still very focused on the estimator itself, not on the prediction.
Can you give me a sense of the nature of the kind of outliers that can happen? I mean, the X's, what do they stand for? Because I always imagine X's are binaries. They are not really, so what are they?
Well, that's a good point, and it depends on the application. So if you're in genetics, for example, the X's will be binary. And so what is an outlier in that case, right? Yeah, so in these, I'm not thinking of that. I'm thinking more of gene expression data, where the X-axis is a continuous variable. Yeah, and
Measurement error that, you know, occasionally
it can happen because of different things. It can happen because of measurement errors, like in the past, like you have a physical slide, and then if you have a scratch or something like. The new methods are different, but still there might be an artifact, like something in the sample went wrong. But it also can be an outlying patient. Like when I work in some of the application, it was like, well, something was completely wrong with that patient, even because it was genetically very outlying, something very different, or that patient is under certain medication that made all their genomic data look very different from the rest. Sometimes they are easy to catch. In particular, the raw cases, they might be easy. The most problematic one are now the cell-wise, because the cell-wise, nothing seems too outlying, but there might be outlying things in the middle here and there. Even those things that are outlying may not be the important ones, but they are still outlying. And it is, and it can be even affected by environmental factors, right? Like at the time that measurements were made, that gene was completely off. And yeah, that's the application that I work on. Now, many other applications can be done. And okay, and no, yeah, and that's because I show you my work, but but yeah, you can. I have in mind to move into generalized linear models. I also have some work done in survival analysis, for example, that is something that is very relevant in the field of biostatistics. But I honestly would like to move more into classification problems. And there are people are working on classification problems, maybe not my methods in classification, but I would like to work in classification problems where the robustness can still be thought in the covariates. So if you think these are my genes, the genes are continuous variables, they can be full of outliers, but now my response is a 0, 1. And so then there can be misclassification there, but not outlying in the sense that I'm thinking of outliers. That is my next step. And one of the main reasons is that most of the problems in biomedicine in general, the response is like that. So even when I'm looking for data sets, or when I talk with my collaborators, they don't have a continuous variable. And I'm like, well, but where did you get this binary? Like it has to, well, yeah, it's because of many things. And because of many things, they just classify the patients into 0, 1, 0, 1, 0, 1. So I really want to move into that direction. I think it will be, as I said in my point, I think it will be used, people will use it more if we basically respond to that need. Yeah, agree. No problem. But there is research in robust statistics for GLMs, okay? I'm not going to say that there's nothing there. Yeah. Yes.
Questions that are very similar to ours, so you can expect them like together.
Ah.
Also, that last point about teaching the undergrad, also need to teach us, and grad, so I think a lot of work, rather, awareness.
Yeah.
So my questions are two basic. The first about outliers. What are outliers? You said if you have outliers, then you may want to use this. So, and so you know, based on the discussion, there was an outlier could be coming from measurement error, so it's literally a mistake. And you would want to, perhaps in that case, go and see, oh, this is a mistake, let's go fix it, or it could be just that by chance you sampled the detail of the distribution. And if you start to then deal with that, you're actually changing. So in one case, there is an underlying truth, and this observation was a mistake, and it's making it difficult for you to get a truth. Whereas in the other one, it seems that you want to change perhaps even the population, right? If you're going to say, no, a person that's in the tail, I don't want them to actually be contributing, even though if I define a population estimate, they would contribute unless truly I set them away. And so that to me seems to be qualitatively different setting.
Yeah.
So, and related to that, if you have I have a question also basic, what is a robust estimator, but we'll get to that in a second. But presumably, if the estimator is meant to deal with these kinds of situations, if you want to get at the population estimate in the limit, in large samples, then do you want your estimator to sort of be less and less robust? Because it is really, it's inducing some bias, right? I mean, if you are going to leave aside some individuals that are part of the population, you don't want to exclude them, but because you want to be robust, you are excluding them in small samples. So do you want to eventually, so meaning, is there a tuning parameter that's determining how much robustness you have? And that tuning parameter needs to somehow dissipate so that you are getting back to consistency, or do you just accept that you're going for a different target?
Okay, so I think the second question is, I'm going to respond to both questions, but because it's closer. Yes, there is the parameter that you tune, and you are saying like, well, this is, I'm going to be robust to 25% of, like 25% of my cases, I'm going to think that they are outlying. And so your estimator is going to be robust up to 25% of your estimator will be robust up to 50%. And that is what I said, the breakdown point of your estimator. So you can tolerate up to 50% contamination, let's say. So for example, the median, we know like you can have 50% of your data can be completely off. It doesn't matter where it is, the median is going to stay at the median. But if you do more than 50%, then the median will be off as well, right? So that is what we call the breakdown point. So the estimator has, when we look at the properties of our estimator, we prove it has a breakdown point of. So I didn't talk about the properties of my estimators, but my estimators have high breakdown point, meaning that I can tolerate up to 50% contamination, or I can tolerate up to that amount. But then the more robust you are, the other properties you're losing efficiency, you're losing other things. So sometimes we say, you know what, I don't want it to be that robust. I prefer that it's 25% robust. And then try to accomplish other properties as well. So in the classical robustness theory, which was before doing variable selection, just the very classical, you have that where you compare the amount of contamination that you tolerate versus the efficiency that your estimator will gain. And when I say efficiency, it's the comparison of the variance of the estimator relative to the variance that the normal model will achieve, like your estimator will achieve under pure normal model. And that kind of ties into the first question that you asked me. The classical paradigm also thinks that there is a distribution, which is your central distribution, and most of the observations comes from that population, and then a small amount of observations are coming from a different population. So you have, but you care about the central distribution. Yeah. It's a mixture.
I'm also Spanish. So because we think the same, they think different. So I think that there is a population that I really care about. Okay, that's the central. And then, which includes tail out. No. The problem is that God is playing that ugly game with you. That you don't see that distribution. You see a mixture of that distribution with some other distribution.
That's why we call it contamination.
You want to estimate the distribution that is the central distribution, not that contaminating. So you are in your mindset, you have a mixture model. Okay, and you care about one component of the mixing, so that's
yeah, I thought.
It's because now she's trying to predict. Now we are in a different game.
Now we're in a different game.
It's like, you have influential points, like some, at least what I understand, some X's will be very influential because if they are outliers, in a linear regression, if you have an X that is far away from the cloud of X's, it's going to really influence your curve, your OLS. Okay? So, but you don't want it, but then when you predict, you're going to be really screwed because you use that X to really skew your regression. I mean, I'm thinking very, very simple, okay? And then you say, no, let me remove that X because it's really screwing me for predicting most of the points, okay? So something like that is what she's trying to do in high dimension. But that's why I was asking, is there some measure of how well your method is doing at really predicting? I mean, because I understand prediction. I don't understand very much variable selection because I don't really understand what is the aim at the end of the game of selecting variables. For what? What is the reason you want to select?
Well, I can respond to that,
Okay.
but before getting there, is the answer to the previous one, okay? I don't take it personal, but the main paradigm is, as she said, it's a mixture. So you have, I was looking for a marker, there's no marker, but 1 minus epsilon of a distribution F plus epsilon of a distribution G. You don't care about G, but you still, the epsilon amount of your observations are coming from G. You don't know which ones. You care about F. You want to characterize F. Okay, so that's.
You don't get sample directly from it.
No, you sample from this mixture.
Points, the tails of that central distribution, they may look like outliers. That's just
They're from a tail. Yeah.
Sure, the components of the mixture for which you
Yeah.
know pretty much nothing. In the old days, they assume only normality for the central distribution,
Yeah.
so it was easier. But if you start relaxing assumptions, it becomes more and more difficult to understand what is an outlier and what is, what belongs to your central distribution. So that's my.
But before you, when we talked about tuning to determine the robustness, you said, well, then it impacts your efficiency and so on. But is that only it? Like if I think of, for example, trim mean, like trim mean will not be, if I set the amount of trimming to a fixed amount, I won't have a consistent estimator, right? For the true mean.
Mhm.
If
it's normal.
If it's normal, but no, I don't
That's
live in
the
normal.
thing, they think normal.
I mean, she didn't say that.
Well, yeah,
It's
she
very
did,
normal.
she said normal.
So, I mean, if you're not in that case, then presumably you would need to let the amount of trimming sort of be data driven perhaps.
Yeah.
You know, and so going to zero as my, if I want to be consistent, right? So that's not really done, or? So that's what I was thinking, the tuning parameter is sort of tending towards eliminating the robustness.
In our case, in the one of the ensembles that we are using some trimming thing, it is data driven, so that we can control both things. But
And you talked about the breakdown point as one criteria. So are there many definitions of what it means for an estimator to be robust? I mean, I'm an ignorant. I just.
yes, yes, there are many.
And so you.
Yeah, there are the infinitesimal ones, there are the finite sample ones, like now we are, yeah. So, yeah, now we are doing a crash course in robust statistics, but yeah. Yeah, to do a course in robust, yeah. But the one that we proved in the paper was the finite sample breakdown point, because it's the one that basically you deal with in practice, right? It's like, how much contamination will I tolerate? I already forgot your question that I said I can answer to that about prediction. The variable selection. Okay, yeah, just this scenario. I was working during my postdoc. We were looking at many proteins coming from a platform where something called they call it unbiased, and it drove me crazy because it's not our bias. So unbiased means that the doctor does not say in advance which proteins they want to look at. They look at the sample, and sample means blood or plasma. It doesn't mean our sample neither. So they look at plasma, they analyze it, which proteins did they see? So it's unbiased in the sense that whatever is there, whatever they can detect, right? But those techniques or those technologies are not very good. They are not approved by FDA. They are not ready to go to the clinic. So they need to know, of all those proteins that they see in the sample, which ones are the ones that they can use to predict something. Let's say, to predict a disease. In my case, it was to predict rejection of an organ in transplantation. Okay, so, and it was to predict the state of the organ. There was a variable that says this patient is going through a rejection, this patient is not going through a rejection. So which are the variables, which are the genes, or which are the proteins that are telling me that this patient is going through a rejection? So I basically want to know who are the key players there that will tell me that the patient is going through a rejection. Big why? Because then I can do like a kit that I can develop in a company, and I can go through an FDA process of approval, and then that will go to the clinic. So for that, I need a variable selection.
Here, I mean, my colleague here has worked a lot in variable importance. He has like measures, right? Can speak about that better than me. But I think that I have to have a well-defined objective function. Like, okay, I want to, I don't know, minimize the mean square error of my prediction with whatever algorithm I'm planning to do, whatever. I mean, it seems to me that just working with a regression and lasso is but one strategy for selection.
It's just one, but it's just one strategy. It's not the only one.
It's a strategy that I don't see it geared exactly to prediction. You see, that's my problem. That you are selecting based on lasso, and it's not clear to me that that's going to minimize the mean square error of what I really want to do. I want an algorithm that you give me a bunch of variables, and I want to select the ones that I want to use in my next study to use for prediction. So I really want to have that set to do the mean square.
Well, but the aim of in particular lasso, it's minimizing least squares of the residuals, and it's based on prediction, and it has a penalized.
Well, that's why I was asking again if there was some analysis
No, it is, but
formalism.
there's no
But
formalism, right? Like, yeah, there might be,
I personally think I want to go to sleep feeling that it's not, that I have some understanding of the math. No, it's like, my obsession.
Yeah. There has been a lot of theory related to lasso, even inferential in lasso, right? So, but I don't know, yeah. But I would say not in the robust version. So at least in the robust versions, no, I would say like, yeah. And then perhaps this goes more towards what you're thinking, which is prediction. I think prediction in robustness is very tricky. And I don't think we solve it. And I think we are kind of like doing like this to many problems, in particular when there's cell-wise contamination. And I'm already detecting many problems in my research, and that's why it's delaying some of the publications, because there are some results that I cannot understand. And I think that the main problem is that we are training things more like in favor of what you are saying, right? Like we are training things more in terms of estimation, but not focus on prediction. The prediction problem is a different problem. To the extent, and I was talking today with Ali about this, to the extent that maybe it's better not to be robust if it comes to prediction. Maybe you will do better, unless you want to predict from the central distribution. Like if your test set comes from the central distribution, then it's very clear. But why your test set will come from the central distribution and not your training set? Like, why are you so unfortunate that you need to train in a contaminated data set, but then you need to test in a clean data set? That's not realistic neither. So when you have outliers in both, which is obvious that you are going to have, the prediction problem, in my opinion, is an open problem. So that perhaps gives you hope that that you are right somewhere. Yeah. Okay. Thank you. No, that's okay. Very interesting question. Thank you so much.
Let's thank our speaker. I'm sure we'll have wonderful discussion still, but yeah.